Today we're going to be talking about the main functions and features of a muscle system, and we're going to start with the functions. So main function of the muscles, of course, is the movement. Skeletal muscles move bones, cardiac muscle moves fluids, smooth muscles move, for instance, intestinal content. Skeletal muscles also maintain the body position, posture, balance, stabilize the joints. They produce heat. That's a really important function of a muscle. Abdominal muscles protect abdominal organs. Smooth muscles form valves, regulate the size of your pupils, give you goosebumps. To recognize terms that are related to the muscle system, we use prefixes such as myo, mes, or sarco. And a really important feature of a muscle tissue is that muscles are excitable. They can receive the signal from nervous system and contract in response. They're contractile, so they can shorten. And they are extensible, so they can stretch and recoil. Let's compare skeletal, cardiac, and smooth muscles. Skeletal muscles are attached to the bones or to the skin. They move bones and or skin, while cardiac muscles are found in the walls of the heart. Walls of the hollow organs, such as blood vessels, intestine, and urinary bladder, contain smooth muscles. Smooth muscles also can be found inside of an eye. Muscles that regulate the size of your pupil or shape of your lens are smooth muscles. The only type of muscle tissue that is voluntarily controlled is skeletal muscle. Cardiac and smooth are controlled involuntarily. And skeletal muscle is the only type that is truly multinucleated or polynucleated. Cardiac muscle has one nucleus. Cardiac muscle cell has one nucleus, very rarely two, while smooth muscle cells always have just one nucleus. Both cardiac and skeletal muscles here have striations. Striations are very small stripes that basically a sign of a particular structure of a skeletal and cardiac muscle and a particular contractile mechanism, which is different from the contractile mechanism in the smooth muscle, which has no striations. Skeletal and smooth muscles cells are not branched, unlike in case of cardiac muscle, which cells are branched extensively. Skeletal muscles can be controlled only by nervous stimuli, which can be easily explained by the fact that those are the only voluntarily controlled muscles. Cardiac and smooth muscles, their contraction can be activated by nervous stimuli, hormones, chemical stimuli, and mechanical stress. Skeletal muscles are the fastest to contract, cardiac muscles are slower, and smooth muscles have the slowest contraction among these three. Now we're going to focus mostly on the skeletal muscle in this conversation. Skeletal muscle moves bones and skin. Every muscle can be roughly divided into three parts: the belly, the middle portion of a muscle that does most of the job, the origin, stationary end of the muscle, and the insertion, the end that is moving when muscle contracts. For instance, in case of biceps brachii, the origin of the muscle is in the scapula. Scapula does not move when we flex our elbow. So the insertion is in the radius. That part of the muscle moves when we flex our elbow. If we know the origin and the insertion of the muscle, we can figure out the function of the muscle, because we can basically shorten it in our brain and just figure out what it's going to be doing. Muscles are composed of very long muscle cells called muscle fibers, and when muscle fibers contract, the muscle contracts as well. Each movement is usually produced by a group of muscles. These groups consist of primary movers. For instance, for flexion of an elbow, primary mover is biceps brachii. It is the main producer of that movement. Synergists, muscles that help in the production of a movement. For instance, brachioradialis is a synergist for the flexion of an elbow. And antagonists, muscles that oppose the movement, muscles that perform an opposite movement. So for instance, biceps brachii flexes an elbow. Triceps brachii, muscle that is opposite to biceps brachii, muscle that is antagonistic to biceps brachii, extends the elbow. Muscles have several anatomical layers of structure, and we're going to start with the connective tissue that can be found in the muscle. So connective tissue in the muscle is necessary to protect it from tear and to attach it to the bones via tendons. The outermost layer of the connective tissue that covers the whole muscle is called epimysium. Under the epimysium, we find the muscle, which consists in turn of multiple fascicles. Each fascicle is surrounded by a layer of connective tissue known as perimysium. Now, each fascicle consists of a bunch of muscle fibers, individual muscle cells. And each muscle fiber, as you can see here, is surrounded by a very thin layer of a delicate connective tissue known as endomysium. And I want to make very clear that endomysium and the cell membrane of a muscle cell are two different structures. Cell membrane is a cell membrane, and endomysium is superficial to cell membrane. If we look at the structure of individual muscle cell, we can see that each cell is wrapped into a cell membrane called sarcolemma, and inside of the muscle cell you can see multiple myofibrils, structures, rod-like structures that contract, and when they contract, the muscle fiber shortens. Between the myofibrils are mitochondria. For ATP synthesis, muscles consume enormous amount of energy. And not pictured here are sarcoplasmic reticulum, an organelle that stores calcium essential for muscle contraction, myoglobin, protein that is responsible for the storage of oxygen, and glycosome. Glycosomes store glycogen in the muscle. Glycogen is the polymer that consists of multiple glucose residues. So essentially, glycosome stores the long-term energy source for the muscle. Now let's take a closer look at the structure of the myofibril. Each myofibril, shown here, consists of multiple sarcomeres. An individual sarcomere is shown here. So sarcomeres are essentially an elementary contractile unit of the myofibril. You can see that if we further expand on the structure of the sarcomere, sarcomere consists of thick filaments, shown here in dark purple. They form a darker band called A band, and thin filaments, shown here in green, that form a lighter band known as I band. So each sarcomere has a dark A band in the center and halves of the light I band on the sides. This explains why skeletal muscle and cardiac muscle are striated. Those ever-changing dark bands, light bands, dark bands, light bands form the striated pattern that demonstrates that mechanisms of contraction in the skeletal and the smooth muscle and a cardiac muscle are the same. So here you can see a further breakdown of what filaments consist of. So this is the thick filament, which consists of the protein called myosin, and each myosin molecule has a tail, which forms a body of the filament, and the head, which can break down ATP and bind to a protein that forms thin filament known as actin. So actin in this thin filament is bound to a protein tropomyosin. Tropomyosin essentially regulates whether or not actin can bind to myosin, and protein troponin that in turn exerts control over tropomyosin. And we'll get back to this sooner. Now, if you would look at the structure of the sarcomere here, you can recognize that when filaments, thin filaments, slide against the thick filaments, move to the center essentially, sarcomere shortens, the entire myofibril shortens. Myofibrils shorten, the cell shortens. When the cell shortens, the entire muscle shortens. So this is how sliding filament model works. Calcium, ion that is critical for initiation of contraction process, binds to the protein troponin. Binding calcium to troponin through several steps causes the binding of heads of the myosin right here and right here to thin filament. And these heads start to pull thin filament towards the center line. That brings the boundaries of sarcomeres, so-called Z-discs, closer to the center line, also known as M-line. The whole sarcomere shortens and muscle contracts. When muscle relaxes, the heads of the myosin separate from actin. Actin filaments slide back from the center line to the sides. Sarcomere returns to the pre-contraction length, and muscle essentially relaxes. What's important to understand that during contraction, filaments themselves do not become shorter or longer. It's the sliding that makes a whole sarcomere to change its size. So how exactly the stimulus is delivered to the skeletal muscle to initiate contraction? So nerves and muscle cells form what is called a neuromuscular junction. It's a single neuron that forms a synapse with one or several muscle cells. So this here or this here are individual neuromuscular junctions. Let's take a look at what happens at the neuromuscular junction. When action potential, the nerve impulse, reaches the end of a neuron, the axonal terminal, that action potential releases neurotransmitter called acetylcholine into the synaptic cleft. Neurotransmitter diffuses across the synaptic cleft and binds to the receptors on the muscle cell, so-called on the motor end plate, and that binding opens the channels, allowing sodium ions to flow into the cell, and that leads to the excitation of the muscle cell and essentially a contraction. So acetylcholine, okay, as we already mentioned, is the neurotransmitter at the neuromuscular junction, and interestingly enough, it's always acetylcholine. There are no exceptions. It's always acetylcholine that diffuses through that synaptic cleft. So when sodium channels are open, as we mentioned before, that leads to cell being more positively charged. We call it depolarization. Depolarization means excitation. Depolarization and excitation generates an electrical impulse that will run through the membrane of the muscle cell, leading into so-called T-tubules. When this impulse, and this excitation impulse that runs through the sarcolemma of a muscle cell finally reaches T-tubules, it opens calcium channels on sarcoplasmic reticulum. Calcium diffuses from sarcoplasmic reticulum into the sarcoplasm, binds to the protein troponin on the thin filament, and essentially initiates a muscle contraction. So here you can see that binding of calcium causes the sliding, contraction, sliding of the filament, shortening of the myofibril, and shortening of the muscle. So this is how the muscle contraction actually occurs during at the molecular level. So first, a molecule of ATP is broken down, forming ADP and a phosphate. Energy that is released during ATP breakdown provides for coking back ahead of a myosin, charging it. Now this coked back head of the myosin has a lot of potential energy. When calcium binds to the protein troponin, right here, it changes the shape of the protein tropomyosin, that long thread. Change of the tropomyosin enables the charged myosin head to bind to the actin. We can see it right here. So finally, myosin heads of the thick filament, are bound to the actin. This structure right here is called a cross bridge. When myosin head releases ADP and phosphate, it pulls the thin filament towards the center, towards the M line. This pulling effort of the myosin head moving the thin filament towards the M line is called a power stroke. So thin filament slides against the thick filament, and then ATP binds to the head of the myosin, which detaches the myosin head away from the thin filament. ATP is broken down into ADP and phosphate, which cokes back myosin head, and the cycle can start over again. And the cycle will continue until the ATP and calcium are present. So I want to focus your attention on one really crucial feature of ATP. ATP is not only a source of energy. ATP is essential for unbinding the head of the myosin away from the actin. In fact, in dead people, after about 12 hours post-mortem, ATP storage is depleted and the muscles cannot relax because there is no separation of the myosin head from the thin filament, and they develop a condition known as rigor mortis. Muscles are contracted basically. Now, each muscle, when contracts, has 3 periods of a contraction. It's called three periods are lag period, contraction period, and relaxation period. And if we go back here, we can appreciate that this part right here, and so release of neurotransmitter, travel of the excitation wave along the sarcolemma, and release of calcium, this will all happen during the lag phase. Contraction phase is here, and then when calcium is gone or ATP is gone, that's going to be a relaxation period. So you can basically depict the graph, the muscle contraction, like this: lag period, excitation, relaxation, contraction, relaxation. Let's talk about some metabolic aspects of muscle fibers. So muscles can be slow oxidative, fast glycolytic, and fast oxidative. I will be focusing on slow oxidative and fast glycolytic, also known as slow twitch and fast twitch fibers. So fast glycolytic fibers are, well, fast. They use mainly anaerobic metabolism, which means they don't need oxygen. And they are producing a short-term, powerful contractions in the exercises such as weightlifting. These muscle fibers are thick, okay? They have few mitochondria and very few capillaries because they don't need oxygen and they don't do oxidative respiration. They white, well, not white, but whitish in color. They store a lot of glycogen because they need a lot of glucose to produce those powerful contractions. However, since they don't need oxygen that much, their myoglobin content is low. They contract fast, but also fatigue fast. Now, slow oxidative fibers are slow contracting and slow fatigued. They have low glycogen storage because they can use glucose extremely efficient in the process of aerobic respiration. But for aerobic respiration, they need oxygen, so their myoglobin content is high. They have many mitochondria and many capillaries. They're usually small, less powerful, and they're red due to the mitochondria and capillaries. And these slow oxidative fibers are essential for low-intensity prolonged exercise, such as endurance exercises, basically running, swimming, and maintenance of posture. Now, what I want to specifically highlight is that the percentage of slow oxidative and fast glycolytic fibers in each person is determined genetically. So if you exercise, proportion doesn't really change with time, or if it changes to a very small extent, but you can change how efficiently you use each type of muscle. It can be very well illustrated if you look at who are the winners of the marathons. It's folks from Kenya and Ethiopia. They genetically are better at running, while the winners of strongman championship are people from Northern Europe, Brits, Scandinavians, Russians. So that explains that they are genetically predisposed to exercises like that. And I'm not trying to say that, you don't need to train, but if you are genetically predisposed to have more fast glycolytic fibers, you will be a fantastic weightlifter, but you won't be a great marathoner. Now let's address some of the anatomy of the muscles. So the frontal muscle on your head is occipital frontalis. Occipital frontalis actually the frontal belly and occipital belly, front and back. So sometimes it's called frontal belly of occipital frontalis. Circular muscle around your eye that allows you to wink is orbicularis oculi, while muscle that allows you to protrude your lips around your mouth is orbicularis oris. Muscle that moves your head, flexes your neck and moves it left and right is called sternocleidomastoid. And the muscle that allows you to elevate your shoulders, see it right here, is called a trapezius muscle. Deltoid muscle is responsible for the abduction of an arm. Pectoralis major is the main muscle that flexes your shoulders. Biceps brachii is the muscle that flexes your elbow. And rectus abdominis is the muscle that allows you to do sit-ups. It flexes your torso. External obliques, so that's rectus abdominis. That's external obliques here. External obliques allow you to rotate your torso and do the lateral flexion of a torso. Muscles of the leg. So the side muscle runs along the lateral aspect of your leg. Is called tensor fascia latae. It's responsible for the abduction of the leg. Pectineus muscle is one of the many muscles that flexes your hip. Adductor longus here, sartorius and gracilis, responsible for the adduction of the leg. Rectus femoris, along with the vastus medialis and vastus lateralis, are responsible for the extension of the knee. Tibialis anterior is responsible for the dorsiflexion of your foot, while gastrocnemius and soleus responsible for the plantar flexion of your foot, basically standing on your toes. Now some more muscles of the arm and shoulder. We're going to look at some different aspects. So we've got biceps brachii and triceps brachii, antagonistic pair. Biceps flexes the elbow and triceps extends it. Deltoid, as I mentioned, it abducts the arm. Vastus medialis, vastus lateralis, and rectus femoris, the muscles that extend your knee. Gastrocnemius and soleus responsible for plantar flexion. Fibularis longus muscle is responsible for the lateral movements of your foot. Some muscles of the back. So temporalis muscle on the side of your head, responsible for chewing movements. Platysma stabilizes your neck, and trapezius is responsible for elevating your shoulders. I already mentioned deltoid, I'm not going to stop at that. Latissimus dorsi is the largest muscle in the human body. It is responsible for the extension of your shoulders. Triceps brachii, extension of the elbows. Flexor carpi ulnaris flexes your wrist, while extensor carpi ulnaris extends your wrist. Gluteus maximus is the extensor of the hip. That's a buttock muscle. You can see vastus lateralis from the side, and these muscles, biceps femoris, semitendinosus, and semimembranosus, that is not shown here, those muscles flex your knee. So those, we call them hamstrings sometimes, and you can see gastrocnemius here, that allows you to stand on your toes.